you sometimes. Hi, guys. Hi. Welcome, welcome, welcome. I'm Looney. That's Llewellyn.
Hello. How are you doing?
I'm good. How are you?
Not too bad. I
You look
love
different,
your hair.
though.
I haven't done my hair.
Your hair has grown. Hey,
No, I
you've
didn't
changed
gel
the
it.
color. Oh,
I didn't gel it.
yes.
You see,
It's
I
flat.
I thought it's a change.
Oh, okay. Yeah.
No, I left my gel at home.
We all. Oh, but we all have changes.
Yes,
Yeah.
how gorgeous is her hair?
What are we doing for the grade 11 today?
We're just going to do exam questions, right? I mean, we're doing exams, so let's put it out there and do some questions, hey, Looney?
All right.
So Looney's brain is here. I've given her notes. She must study, and hopefully she answers the questions.
No.
Look at those eyes. She's a really
You see,
good
I was
cross-eyed.
rolling my eyes. Guys, you know he sometimes puts me on the spot. But I'll try. I'll try my best to answer. But you know my answers. They're not life science related.
Of course.
They're something else. But I hope you guys do enjoy the show. Remember to hit us up on Facebook, on facebook.com/LearnExtra. Our Twitter handle is @LearnExtra. And you can get the show notes, the videos, and the schedules on learn.mindset.co.za.
You on form.
No, but I wasn't fast, hey.
No, that was perfect.
Exactly.
That was
That was
brilliant.
for you. You see that pace?
And
tried.
the winking.
Yeah,
what more could we
everything.
want?
Yeah.
Right, let's go do some work.
Cool.
Right. So we're gonna have a look at exam questions. Right. So first thing we're gonna have a look at, of course, we need a challenge question. Hey, Looney.
Yes.
We need to challenge you guys out there because what's life without a challenge question? Hey. So something that you can do, and then give me the answers, and then I give you the answers afterwards. So let's have a look. Okay, James and Peter. What we actually did was James and Peter wanted to do an experiment on photosynthesis, right? So what they did, this is what this says over here. Okay, they took this lamp and they put it in front of a what's that thing called? I must just get the pen to make sure you guys whatever. They submerged it into a. See, it's too long ago. James conducted a photosynthesis experiment,
What are you
and they
trying
used
to
the
say?
lamp shining onto a gas jar. That's what I'm looking for, a gas
Oh.
jar.
I was like, just read. Gas
Yeah, I
jar. I was
know.
wondering, what are you
Gas
trying
jar.
to say?
Anyway, so they put it in the gas jar, and they filled this gas jar with water, right? And not only did they put water in there, because there's specific things I'm looking at. That's why I'm showing you, okay? So it's a gas jar we're looking at. They put it with a lamp. There's a lamp involved, okay? Then what they did was they put a plant, but in this gas jar, and they filled it with water, okay? So this plant is inside the water and it's submerged in it, okay? So there's a plant in it. So a water plant, well, let me see here, containing a water plant that is submerged inside it, right? And not only that, they took a sodium carbonate, right, a solution of sodium carbonate, and they added it in for good reasons, right? So they added this sodium bicarbonate in, right? Then sodium carbonate, not bicarbonate, sodium carbonate. And what they've done is they've put this thing here, and they're moving the lamp closer and pulling it away in different intervals, right? And as they're doing that, they're recording something, because remember, James noticed that there's bubbles coming from these leaves, okay? Now, that is what you guys will see on, it is, where do we post this? On Facebook, right?
Yeah.
Yeah, so we've got it on Facebook. But now, because you're watching, right, I thought I'd give them a picture as well, hey?
Okay.
How cool is that? So that is what you will see. Now, just because I want to cheat, like slightly, because cheating's not always bad, right? So what I've done is I've put a picture on it. There it is. You'll see the bubbles coming along over there, and there's the lamp and everything, okay? Now, the questions are very simple. I'm not going to answer them, right? Here we go. First question. What is the aim of the above experiment? Okay, what is the aim? What is the purpose of this experiment? That's what they're asking. It's the first question. Second one, what is the function of the sodium bicarbonate? Okay. Third one, name the gas released during, so we want the gas released during the experiment, so those little bubbles that are coming from this plant. Right, next one, explain a simple test that can be done to confirm the presence of the gas mentioned. Now, when you do an experiment, there's certain things that you've got to do after the experiment to prove your theory, okay? Like, if you remember, we did, I don't want to give it away, so I'll leave it in the back at the end, okay? But you've got to be able to do something to prove what gas that is, okay? And then I've got another one here. It says, Name any 2 environmental factors besides, okay, that's important, look at that word. They're telling you they don't want certain things. And the things that they don't want is the light intensity. Sorry. They don't want the light intensity, which is very, very important. So you cannot mention light intensity. Okay? They want another 2 that could affect the chemical process shown below. So what is happening, or the making of whatever. Okay? They want to know what else they can adjust to make it go faster. Okay? And then one more. What conclusion can we arrive at from the information supplied above? Okay, so what can we get from this information? We know this experiment. You have to study it, you have to know it, right? And at the end of the year, you're going to do a prac. Even now, you're going to be doing a prac. So make sure that you know how to do these pracs. Am I right, Looney?
Mm-hmm.
So one of these days, I'm just going to rock up here and say, Looney, do the prac, and she's going to look at me as if I'm mad. Yes.
No, I'll do it.
Good girl.
You know, I'll try.
Of course you'll try. You do everything with me.
Yes.
That's why we love each other so much. Right. Now, let's go on to the questions. So I hope you've got that. Right. Now, all I need you to do is do that, and let's go on to questions where I can have a look at it and help you out. Right. Remember, we started off right in the beginning. We looked at body plans. Can you remember that? That's animal diversity. Am I right, Lenny?
Mm-hmm.
Okay. So what pictures do you see, Lenny? Come on.
A crab.
A crab. Yeah.
Oh,
Ah, you see, she's beautiful.
what is that?
That's a sponge.
A sponge.
Yes. The thing you wash with.
No, man.
It's almost like coral.
Oh, which one? B.
B, yeah.
And then C is a jellyfish. No.
It's the same family, yeah.
Oh.
It's very nice, almost. Yes, it's almost like the same family. Very nice. That's
Okay.
good. She's getting there.
Trying.
Slowly but surely she's getting there. Right. Now, let's have a good look at the question. Bring this up here. Okay. What kind of symmetry? What kind of symmetry? And what does symmetry actually mean? Lenny, can you remember? What does symmetry actually... Look at that. Wow, that is perfect. See, she listens to me. What more could I want? Exactly. So now she wants to know what symmetry. So let's have a look. It says, what symmetry does each of them have? Okay, it's simple. Firstly, I can cut this one. Can I cut it this way? Can I cut it that way?
Mm-mm.
There's no way. It's not going to happen. Right, so I'm going to erase it. Okay? Because I want to make sure you can understand it. Now, the only way I can cut this, if I have a look, is right down the middle. Whatever's on the left is the same as on the right. Right, Lenny?
Yes.
Cool. This one. How am I going to cut this? Where can I see? Because remember, symmetry has got to do with exactly what's on the left and is on the right. Okay? So, if I cut this down the middle, is it going to work? No. How about that way? Is it going to work? No. That way, that way, that way. Not going to work. None of them can work. So this whole thing is wrong. Okay? It doesn't work. So there's no symmetry there. This one, on the other hand, this is quite nice. This can be cut that way. It's the same. That way, it's the same. That way, it's the same. And that way, it's the same. Right? All symmetry. Okay? So if I have a look at this, this is bilaterally symmetrical. Or this is bisymmetry. This is asymmetry. And this is radial symmetry. Okay, does that make sense? The one is cut down the middle, left and right. Okay, one you cannot cut because it's always going to look different. And one, no matter how you cut it, you're always going to have the same. It looks like the same on all the sides. Right, we all understand. That's quite nice and easy, okay? So, let's see if we are right. First one, A is bilaterally symmetrical. Right, then you get asymmetry, and then you get radial symmetry. Right, very nice. That's very, very simple. What animal processes? Okay, so what animal processes happen in these three things here? Okay, in these animals. What do they do? Why is it so important for it to happen? Okay, in C and A and B. Okay, they want to know exactly what's happening. So, here we go. If I touch that, okay, these animals are usually, if we look at A, these animals are usually very, very movable, okay? And they do complex movements, right? And not only that, they've got this backbone in the middle, or they don't have to have a backbone, they've got this nervous system in the middle to do complex movements. That's A, okay? A is very important. We're looking at A and C, okay? A It's very important. It's got this central nervous system, and in that central nervous system, there is a makes the muscles move in certain ways. For example, flying, okay, or crawling, or walking, or whatever. So if we have a look at it, these animals usually move. Oi, that's not a good thing. Let's do that rather. They usually move as a central nervous system. Right? Can you remember that? Okay, they are doing very complex coordination, like, here we go, flying. Very important. Okay, and sea animals are there. So in other words, animal C, if I just move this up again, animal C is on the ground, and what it wants is it wants to see all the way around. Okay, it doesn't need to move. It's going to stay there, but it wants to notice its whole surrounding, so it can see right round and all over the place, right? So that's why they're so important, and that's why they look the same all over the place. And let's just make sure that is the answer. Okay, it says there, allows animal C to view the environment equally from all sides. Very important. Okay, it catches its prey that way, that specific animals. Right, the next one. If you've noticed, I've put the question in the middle of what's the answer in the middle. That's not a good thing. It happens at times. Sometimes it happens, sometimes it doesn't. Okay, so if I look at the next question, now we're going to get into the things that are a little bit more difficult, right? So this is where I need Looney's help. Am I right, Looney? See, now she starts cringing. Okay, which is quite nice because I like Looney to look at me because I feel special when she looks at me. She gets that smile. Right, so if we have a look at it, look at that smile already. Right, different animals. This is a phylogenetic tree. You know what that is, hey, Looney?
Of course I do.
Of course you do. You can laugh in your teeth. That's so perfect. Right, a phylogenetic tree. That thing there is very important because it shows you where things come from. Right? It's evolution almost. Okay, so it's like we all come from one thing and we come up and we split to different parts, and that's what we're looking at, right? So if we have a look at this, we have, just look at this picture, right? We've got the ancestral protista, right? Protists, there they are. And then what happens is they moved up and they split into different animals. And if you have a look at it, we have the Porphyria. Well, I'm not going to do that because if I do that, you can't see the name, right? We've got the Porphyria, you've got the Cnidaria, you've got the Platyhelminthes, you've got the nematodes, you get the mollusks, annelids, arthropod, and then your chordata. Those are the ones that we're looking at that's most important. Right, so those are the ones we're actually going to have a look at. Now, they ask you questions on this. It's quite nice. And the questions that they would ask roundabout is, according to the phylogenetic tree, which group, well, let me underline it, according to the phylogenetic tree, which group is the ancestors of, so there it is the ancestors of the animal kingdom. So they want to know, who was the first one? Where did the first one come from? You understand what I'm saying? And thank goodness Looney has a clue. She does have a clue. And what would you say? No, stop laughing.
No, man, I'm trying to. Let me see the picture.
Okay, there we go. If we have a look at the picture, there it is.
Which group was
Where
the
did
ancestors
they come
of?
from?
Oh, protists.
Oh, that's a great answer. So that's exactly where they come from. Your protists. Okay, so they come from your protists. That's where everything started. Now remember, your protists are your most simplest animals you get. Okay, they're the ones that they are simple and they don't have to stress. And you understand? They're not complicated, right? Like Looney and myself. More Looney because she's very complicated. Right, me. If we have a look at the next one, how many animal phylos are shown on the tree? So in other words, at the end of everything splitting, how many different types of phylum do you get? Now remember that this is the present day phylum. Okay, and they want to know how many are there. Okay, it's not that difficult. I showed you. Remember I told you about the annelids, the platyhelminthes, the chordatas, and all of those? Those are what we're talking about. So this is simply count. Okay, so let's have a count. That's 1, 2, 3, 4, 5, 6, 7, 8, and of course 9 starfish. Right. So there's 9. So Looney, how many different phyla are there?
9.
9, good girl. It's very easy and very simple. Right. Now if we look at the next one. Okay, the first major split, so the major split, if I can get a pen going, the first major split in the animal kingdom was into radial and bilateral symmetry. That's simple. Can you remember when we did this whole thing? We started off with symmetry, and we found out that we all started as a single cell or whatever, and we went from the protist, and then we split up into asymmetry, symmetry, or radial symmetry. We went from asymmetry to radial and to bisymmetry, and then we started going in through to triploblastic and diploblastic, and we had a look at what are those things called, your backbones, and we had a look at there's one I'm missing. I'm going to leave it out, okay? And I want you to tell me after the break quickly. It's a certain thing where you have a false one and you have a true one. Looney, can you remember what it is? Okay, it's a difference between a platyhelminthes and an annelid.
Don't
big
remember.
words. So what I think we need to do, I think we need to go for a break before I start answering these questions, right? And then, when we get back from the break, let me see if you can give me those answers before I give you mine. Right.
Okay. Yes. Yeah.
All yours, girl.
Mine says let's take a very quick break, and I'll see you straight off the bus. Welcome back, guys, from that break. A quick shout out to Andy Swa, and she says she wants to say hi to all the grade 11s of Mahlabatini High School. So
What about us?
Hobby, huh? Oh, and hi to us. I'm saying
you
hi
say
to
that?
us. No, I'm just saying hi to us. Hi.
I thought we loved.
We are, man. What are you
Just
talking about?
putting it out there.
We are loved, but she's just saying hi to her friends. Yeah.
Good.
Yes.
Now, before we went on the break, I asked you to tell me what's that thing about that body cavity where we can put a stomach and small intestines and all of that. Sorry, Looney, did anybody answer that?
No. Hey.
Nobody.
No, but I think I know.
You know. Okay, let's hear
Yes.
this.
A silo.
Very nice. You get a coelom. That's with us. You know the chordatas and everything? It's that coelom where the bag is. Now, the question is, what do you call one that is false? That's false. Come on, Ludi. If you can get a coelom. The other one's a little bit more difficult.
A pseudo silo.
That's it. Pseudo coelom. You're
I'm
telling me
clever.
she can't get the stuff.
So
I think
clever.
that was brilliant. Huh? You weren't on the internet, were you?
Never.
I'm glad to hear that. Right, so she's right, but we'll get there. Okay, firstly, which phylum does not? Well, let me do that line again. Which phylum does not form part of this split? So remember, I said to you they split. We had one, and then we had bilaterally symmetrical and radial symmetry, and there was one that didn't. So if we have a look, there's the first split over there. Okay, so if you have a look at it, there's the first split. One went that way and one made the rest. Now this one that made the rest is the one we're looking for. It is your porphyria. That is what we're looking for. Okay, and if we have a look, porphyria is the exact answer we're looking for. That is asymmetrical. It's your sponges. Right, now let's go to the next one. Pen, very good. Which phylum has, radial symmetry? If we go up, which one's got radial symmetry? So, will it be a worm? No. Would it be this mollusk? Those shells that are on there, are they all perfectly, you can cut them in any direction? No. Can it be a crab? No. And can it be a chordata, like a fish or a human or anything? No. The only one I haven't mentioned here is your Cnidaria. Okay, that is your things like your jellyfish. Can you remember your jellyfish? It's got all the tentacles all around, right? It lies like this. It's got no brain, and you can cut it anywhere. You go up to on the beach one day, and you have a look, and this jellyfish comes along. Can you say which way you can cut it in that's not going to be even? No, it's radial. It can be cut any single way. Am I right, Looney?
Yes.
Do you know what? Have you seen a jellyfish, Looney?
A jellyfish.
A jellyfish.
I think I have, but it was out of the water.
Exactly. Of course. Thank goodness, because you know how they sting you.
Sting.
Yeah, they got little stingers on them.
Jellyfish.
Yes.
Really?
Yeah.
And they can kill you. No.
Depends which ones.
Oh. Really?
Yes.
Oh, I didn't know that.
Well, thank goodness I told you. So if you see it says there, warning, jellyfish, don't swim. Try it. It stings. It's beautiful. So we got to...
It's beautiful.
Yes, Cnidaria. They have stinging things for a reason. Like a blue bottle. Have you ever been stung by a blue bottle?
Blue bottle.
You see, these people nowadays. Right.
What's
it's
a blue
sting-
bottle?
Durban Beach. It's got beautiful blue
I've
bottles.
never been to
And
Durban.
if it stings me and I go touch you, that stuff stings you as well.
Really?
You got to put aloe leaves
Oh.
and vinegar and all of that to get rid of it. Right. So there's some useless information that will help you one day when you're at the coast. How nice is that? Right. So let's have a look at the next one. Okay. Very simple. The second split is animals which have coelom. Right. And animals which lack a body cavity, which we mentioned. Right. Now, the question is, which phylum has no body cavity? So which phylum has no body cavity? So what I want to do is, I just want to bring it down here. Which phylum has no body cavity? So let's have a look. Coeloms go up, okay? And no coeloms go that way. So which one has no coeloms, right? It's not a difficult one. It's quite simple. It is your Porphyria, okay? There it is. Porphyria has no coeloms, because remember, it's going up to the... Where are you going? Come back up here. Okay, it's got no coelom. If we go to the next question, a body cavity. Remember, a body cavity is that whole part on the inside. If we look at the next one, what does it mean? I'm busy reading. Which phylum has no body cavity? And a body cavity, as I said, is what's on the inside. Okay, where is it? Which means it does not have a true body cavity. Hey? Then, come on, there we go. Platyhelminthes. So, which phylum? Here we go. I just want to underline it. Which phylum has pseudocoelom? Remember, I asked Looney, what's a false coelom? A pseudocoelom, and that, of course, is your Platyhelminthes. The nice way of looking at it, it's very simple. Is pseudocoelom is this big word. Am I right, Looney?
Yes.
Okay, and the flatworm is also this big word, Platyhelminthes. That's a nice word, hey? You all want to sound clever? Mention Platyhelminthes.
Okay.
Right, you walk along and speaking to your mates and the Platyhelminthes was cool. Hey, how nice does that sound, eh? It was a beautiful night. The Platyhelminthes
But who speaks
was all
like
over
that?
the place. Hey, you'll sound smart, but you're not going to know what you're talking about. But Platyhelminthes...
Can I interrupt you for 2 seconds?
Yes, with pleasure.
Just, oh my gosh, now it's gone. Okay, I'll interrupt you.
Just
It
now.
was a question that's related to what you're doing, so I just wanted
What,
to...
with the
with the
Platyhelminthes?
pseudo...
Oh, pseudocoelom.
Yeah, pseudocoelom. What's the difference between...
A coelom
Yes.
and a pseudocoelom.
Yes.
Okay, very simple. A pseudo and a pseudocoelom. Let's see. A body cavity, if I've got my digestive system runs down there, right? It's very simple. Your digestive system runs down there. So what I'm going to do is I'm going to turn it like this, okay? So that you can see. So that is your digestive system, okay? Now, your pseudocoelom, right, is going to be a false coelom. Okay, it is, how do I put this? It's going to be part, I'm trying to think now how am I going to explain this to you so it makes it easier. It's going to be a whole part. So this is going to be your body, okay, your mesoderm, okay, this is your endoderm, okay, and this pink stuff here is a bag, and it's completely all the way around. And that is called a false coelom because it goes the whole way around. Okay? Now a pseudocoelom, okay, if I do the same thing, if there's the hole, very nice, okay, a pseudocoelom, you've still got, wait, let me go back, you've still got your mesoderm over there, and it goes all over the place, right, there it is, okay, but, let me erase a small piece of this, okay, there we go, but your pseudo comes out, and it goes with it, and it comes back, and it doesn't form, the mesoderm still comes through there. So it's not a complete round. The mesoderm is inside here, so the mesoderm is this whole piece here, as well as on the inside between the endoderm. Can you see the difference? Look here. The mesoderm is right by the coelom is right by the endoderm. Here, the mesoderm is on the inside of the coelom and on the outside of the coelom. Does that make sense? And this bag here, this bag is where you're going to put your organs, like your stomach, like your heart and your liver and all of those things. Make sense?
Thanks. That was from Asipe.
From?
Asipe Khadebe.
Asipe
Khadebe.
Khadebe. I like that one. Khadebe. I can't say his
No,
first
it's fine.
name.
It's fine.
I would try. I'm going to try. The end of the show, I'm going to try. Okay, so hopefully you understand it now, the difference. Okay, the next one is very simple. Okay, it's from the diagram, identify 3 phylums. Oh, half of the question's gone again. Okay, let me just push it. No, man, let's not start this. There we go. 3 phylums, it's supposed to say. 3 phylums that have got a coelom. Okay, so it's 3 phylums that have got a coelom. And it always starts with the roundworm, because the roundworm is the most simplest, okay, the easiest with a coelom. And then it goes all the way up to the chordata. So we're first going to have a look at, you're going to get your annelids, right? If I can get a pen going here, it's your annelids. If I still can get a pen going here, it's your annelids. It's your arthropoda. Remember, your arthropods are like your, let me see, Looney, do you know what an arthropod is?
Yeah. Arthropod.
Yeah. Come on. Right? It's got that funny legs and it can crawl on you and it sometimes
A spider. No.
Yes.
No.
No, but a spider is part of it. Yes.
Okay.
Spider's definitely part of it. And then you get a bee.
What are you talking about? It can crawl on you.
Arthropods have got an exoskeleton of chitin, right? It's that you know all your insects?
Yes.
Your insects, all of those things are your arthropods.
So a spider is an arthropod.
Yes, of course a spider is one of them.
Come on.
Come on. That's the first thing. I know who's scared of spiders. I must bring one, right? And then, of course, your chordata, which, of course, Lunia, give me an example of a chordata there, my sweetie.
A what?
A chordata.
Your head.
My head.
You're pointing here like he's going like this.
Yes. What is a chordata?
Your head.
You the same. Okay, here we go.
People.
People. Chordata. Sorry.
Don't do this. You should have said me or something.
My head. You're talking about whole animals here and my head. We're only talking about the head of a Platyhelminthes.
My answers are not life sciences related.
You've got to love it. Right. Now let's look at the next one. Okay. This is quite a nice one. The following table shows the effects. Oh, this is quite nice. Now we're going on to things that we did before. You know, things like viruses and bacteria. Right. So we always talk about this thing called HIV virus. Right? Human efficiency virus. Right? The HIV. Am I right, Lily? It's a big thing.
Yes.
Okay. Now there's this drug. Right? It's actually called PEP. Okay. So at the bottom, I've got a table of showing you when they get PEP when a certain amount of time after they've been infected with HIV. So here we go. The following table shows the effects of post-exposure prophylaxis. Okay, that's what PEP stands for. Drug. It's a drug. They give it to people, right? And the effects, okay, how it affects after different periods of exposure to the HIV virus. Study the questions, study the table and answer the questions that follow. So what it's actually saying is, okay, the period, okay, there we go. So it's the amount of hours that they've given it to the person after being affected. Okay, that's what the period is. Then, of course, how it affects effectiveness in 100% in percentage-wise. So if I have a look at it, if you got it now and you take the pill now, immediately, it works 100%. Okay? After 24 hours. So, for example, you prick yourself with a needle by accident that somebody else has pricked and they had AIDS, or HIV should I say, and you prick yourself, and then you go to the doctor 10 minutes later, it will still work. It'll get rid of it. Okay? Then 72 hours, okay, it works 52%. Okay, 52%. You've got a 50-50 chance it's going to happen. And then anything under 76 hours, right, it's not going to work. There's no ways. Okay, it's not going to help anymore. You with me so far? Luni, you with me? Good girl. So that is what's happening. Now it says here, draw a simple line graph to show the above information. Now, before we start with a line graph, or with a graph, I just want you to remember, you get three main types of graph that we're looking at. We're looking at a histogram, we're looking at a bar graph, and we're looking at a line graph. Okay, they tell you use a line graph. With me here. A histogram and a line graph work together. A bar graph is different. Okay, a bar graph is different information. Like, for example, I like using rainfall. If I say to you, rainfall in Johannesburg from January to December, okay, rainfall in January to December, in Johannesburg from January to December, it's in Johannesburg, right? So that means it is one after the other. So it's going to be a histogram. They're going to touch, the information's joined together, right? So in other words, that graph would look something like this, right? Okay, they touch. A bar graph looks like this. I always say it, that a bar graph looks like bars in front of you that you can grab. You can stick your hands around the bars. That's why it's called a bar graph. Okay? And a bar graph is when the information doesn't join together. So in January, the rainfall in Cape Town, Durban, Eastern Cape, do you understand what I'm trying to say? They're all different places. So this one doesn't affect this one. Make sense? That's a bar graph. And of course, a line graph and a histogram work together. Okay, just putting it out there. Now, to help you with that, next thing we're having a look at, we need to have a look at the thing called the x-axis and the y-axis. Which is which? You've got the dependent and you get the independent. The dependent goes on the y-axis. The independent goes on the x-axis. Okay, x is at the bottom there and y is over there. Simple. Right, we're happy with that. Now, dependent. The one is dependent on the other. So, let's see. Which one is dependent on which one? Okay, which one is dependent on which one? So, here we go. This is what the graph looks like. And I actually want to give you some more space. First of all, on the x side, period, where's this thing? Period of exposure. Okay, and then of course, this one is dependent on how long ago you were infected. Okay, this, you cannot mess around with that. Let's just get rid of that because that's not nice. We don't like drawing over our stuff. Okay, this one's not dependent on this one. Okay, period of exposure. So because it works 100% because you were only exposed there. Do you understand how it works? The one is dependent on the other. So that's the first thing you look at. Which is the x-axis and which is the y? This is the dependent. This is the independent. You happy with me so far? Okay. Then you need to give me a heading. People forget to give a heading. Luni, how important is a heading?
It's very important.
What is the first thing you do when we start the show? What is the first thing you do?
A welcome.
Hello, and then you say, I am
Luni.
And I am
Llewellyn.
There you go. So you are giving who we are.
Yes.
It's important. So what you're doing is you're telling me what this is. You need to tell me. If you do not tell me, I have no clue. You have to give me a heading, right? Okay, that's the next thing. Make sure that it goes up evenly, 10, 20, 30, 40, 50. Okay, you get marks for it. Make sure you put the dependent and the independent variables in. It's very, very important. Okay, and please remember, make the lines clear. Take time on a graph. It's easy marks. Okay, it's very, very easy marks. So relax and take your time with it. Okay. I think I need a break. What do you think?
Yes.
Right? Because I'm stretching your brains. Okay? Please don't forget the challenge question.
All right.
Because when we get back, I'm going to start with that. How's
What's
that?
this? Because
Put some effort into it, hey?
okay.
Well, it's the same as you doing.
No, it's fine. Really. Mindset says we are going to take a break, but before we do, congratulations to Lakira Tao. You have won yourself a Casio calculator. Hope you enjoy your prize. It will be coming to you very, very soon. And Mindset says if you want to be like Ulaki, all you need to do is enter the Test Yourself competition. How you do that? The link is on our Facebook page. You click on the link, fill in all your information, answer the questions, submit your entry, and entries close on Friday in the afternoon at 12. So make sure you enter before Friday, 12 noon. Alright. Let's take a very quick break and I'll see you straight after this. Hi guys, welcome back. Please, no, don't do that. Now I'm trying to think what I wanted to say and you're laughing. Oh, yes, mindsetters. You see, when you do shows with Llewellyn, sometimes your mind just goes off track. Now it's back. If you have any questions, guys, and we don't get through all of them by the end of the show, we do have 3 types of help desks where you can send your questions. The Mixit app, the Facebook app, and the Learn Extra Help Desk. And remember, the Learn Extra Life Sciences Help Desk is proudly brought to you by Excel. So please make use of it, guys. Thank you. I'll check for you the questions. You can just carry on.
That is awesome. You know that I've got I'm already at home, and then I get these questions with this and then I've got to answer your questions. So please don't think that I'm not helping. And once I leave here, that's it. No, I'm there
carries
for
on
you all the time.
All right.
Okay, challenge question. I know
First.
you guys have done it. Of course.
Okay.
That is first.
All right.
And then we'll get to these questions.
Okay, cool.
Right, now, before I go on to the challenge question, please remember, the only way that you're going to get good marks for life science, and this is a secret, okay, you won't believe the secret, is have fun.
Yes.
Okay? It's about having fun. If you mess around with things and you touch things and you play with things, you playing makes you remember, right? If you don't play with something, you have to study 10 times harder to get it, right? Go and play. Now you know why I brought the digestive system out here, because it's good to play. You saw Looney's face, her eyes lit up. She knows that the large intestine absorbs water now. Am I right, Looney?
Yes.
Because of the different feces that we took. Right? She remembers the villi because she was telling me about it and the increasing in surface area. And it's all about playing. You've got to play. If you talk about, like with the grade 10s, they were doing bones and all of that type of thing. You've got to play. You've got to look at joints. You've got to enjoy it, because that's the point of life science. It's about life and nice things. Play. The more you play, the better. Always. Hey, Looney? That's why I'm always smiling and laughing, because it's supposed to be fun.
Yes, you must have fun with learning. You
I
can't
would
just
jump up and down,
Yes.
but my knee won't allow it. Right. So, now, the question was, this time I'm going to let you have it, okay? It says, Peter and James conduct a photosynthesis experiment, okay? They use a lamp to shine onto a gas jar containing a water plant, submerged in water. So simple. They add a solution of sodium, it's supposed to be bicarbonate, right, to the water. They add it to the water. James notices bubbles, very important, of the gas emerging from the leaves. So he notices bubbles coming from the leaves, okay, of the plant. During the experiment, the distance between the light source and the apparatus is changed to at regular intervals. What does regular intervals mean, Looney?
regular intervals.
Yeah.
Like regular intervals.
Like? Exactly. You have regular intervals of subjects. So you have, some people have a 45-minute lesson
Yes.
of English, then a 45-minute lesson of science, then a 45-minute. That's regular intervals. It's
I
exact
couldn't
same
explain
time.
like regular
Yeah, it's
intervals. I
regular.
can see it
This
in
is-
my head. I can, yeah, but I can't explain. You explained it
But
properly.
that's good, Lily.
Yes.
You need to know what's going on. Right. So this is what it looks like. And what they did, they counted the bubbles. Very important, right? They counted the number of bubbles released at various distance, okay, and they recorded it. So what they did was, let me just put this there again. There it is. And as they put it here, they count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and they count all the bubbles for that certain amount of time. Okay, and they find out how many bubbles are produced. What they could actually do as well is they stop it. So if they put it here for 10 minutes, when they finish, they stop or they catch all that bubble, all that gas, and they see how much gas is produced. Right, so that's the whole point. They want to see how much gas is produced at different intervals. Right, so the first question, if you can remember, right, I'm going to hopefully, it's been covered both, so humor me here. Okay, the first question was, what is, let me get the questions going here. The question was, what is the aim of the experiment? Okay, the aim of the experiment was very simple, to see if light intensity increases photosynthesis, which of course we know is going to happen. Okay, so it says, the intervals that affected the light intensity on the rate of photosynthesis. So does the investigation, does the light intensity affect photosynthesis? Of course it does. Okay, it's very important. Okay, now, next question. Oh, let's go. Next question, it says, what does that sodium bicarbonate do? Okay, the question was, what does it do? Why did they add it to the water? Lini, can you tell me? Why would they add sodium bicarbonate? What do you think it would increase? Think about it. Just think about it. No. Come on, think about it. Okay, let me help you.
What was this?
What is that? What am I breathing out?
Carbon dioxide.
Good girl. So it increases carbon dioxide. You see, I give you the easiest clue, and she cannot even
I was
put
like,
one and
what
one
are you
together.
doing? Okay, sorry, yeah, carbon
Come on,
dioxide.
help me out here. And you answer the question. Yes. Right. So what it does, it increases the carbon dioxide. Right. It increases the carbon dioxide inside the water. Now, why would we want to do that? Right? We all know that to go through photosynthesis, if you remember carefully, to go through photosynthesis, you're going to notice that you've got to increase carbon dioxide because you get a light phase and you get a dark phase. And inside the dark phase is where we use carbon dioxide to produce glucose. Can you remember that? Right? So we need the carbon dioxide. So the more carbon dioxide we put in, the better. Right. Now, the next question. Okay? Just mind me, I'm opening it up so we can see what's going on. Now, the next question was, that gas that was produced, what gas is it? Okay? Now, we know photosynthesis. Let's see. Okay? With photosynthesis, we're going to take in water, right? Because we're pulling it from the leaves, right? We're going to take in carbon dioxide. Can you remember? We're going to take in sunlight. Yes. And what are we going to give off? We're going to give off water vapor. Okay? We're going to produce water vapor. And we are also going to produce what, Luni? Caught you in the act. What does a plant, what
Oxygen.
gas, yeah, you see. What gas does it produce?
Oxygen.
Very nice. It produces oxygen. It helps us breathe. So remember, we give off carbon dioxide. They take in carbon dioxide. They give off oxygen. We take in oxygen. Right? How cool is that? So that's what it's going to produce. It's going to produce oxygen. There we go. Okay, well, let me just get a pen. There we go. Okay. Explain. Here we go. Explain a simple test that can prove what gas this actually is. Now, Lini, you did life science in grade 8 and 9, am I right?
What was it called? Social? What was it called? Social?
No, social science was like history and geography.
Oh.
We're talking about NS here.
Oh, natural science.
That's
Oh, my
good.
word.
Yeah,
Back in the
those good old
day.
days. Okay, now, how do you test for gases? There's two gases we're going to test for. Oxygen and carbon dioxide. You think? Yeah, here we go. Okay. Now, as a science student, you should also know this, right, Lynn?
How you test for gases?
Yes. Now, the first one we're going to have a look at is carbon dioxide. Now, if I remember carefully, you take carbon dioxide and you pass it through the stuff called lime water, and it turns the lime water milky.
Oh, I remember something like that.
There we go. Can you remember we used to do
Yes,
this?
yes, I
You put
remember.
this lime water, see through, you go with a butt, and it goes all milky. It was so
Yes.
cool. Right, so that is to test for carbon dioxide. Now, how do we test for oxygen? And the nice thing about this is, even if Looney does not remember her grade 8 and grade 9 life science, she should remember the other day I did the experiment.
Oh, that
so
day.
What that day? And I took something and I proved to you that it was oxygen. What-
You let it was a stick. You lit it up,
Yes.
and then it was orange, like it
Yes.
was burning, and then you what did you do? There was water, and then
No,
you
there was no water.
Yes.
No,
Oh,
that was
okay,
to get
that
the
part.
oxygen,
Okay,
right?
you got
And
the
then what did I
oxygen,
do?
and then you dipped the stick in, and then it
Went
it's
into
got
flame again.
yeah.
Right. And that stick is called a glowing splint. It's very
Yes.
simple. You put in a glowing splint and it'll burn much brighter. That was very good, Looney. So you do concentrate. But
I
then again, I
do.
always involve her in the experiment. So see what I mean about playing? Hey,
Yes.
Looney?
So I remember.
Exactly. You play. You enjoy life science. I mean, even if you find things boring, and I know people don't like much plants, go and play. Go look. It's a lot of fun, right? And there we go. The answer would be a glowing splint, right? Brighten. So when you put in their mind burst into flames, there was so much oxygen, right? Now, you put it in a test tube, so he would catch all the stuff, and you put a glowing splint in it, burst into flames. That's the whole point of it. It's quite nice. It's a lucky experiment. Okay, now the next one. Name any two environmental factors besides the light intensity that could affect the chemical reaction. Okay, now let's think about this. What else can we increase? Looney, when you have to do sport, you remember sport, eh? Because
Yeah.
photosynthesis is like sport for the plant, right? Now, if it was very cold, how much do you feel like doing sport?
No.
And as it's getting warmer,
You want to go out.
you want to
more.
do better and
Yes.
better. Now, so what do you think? It would be heat, right? The hotter you become, the more you want to do the sport, right?
Oh, yes.
So the hotter you become, the more you want to, the plant wants to photosynthesize. But wait, there's a catch. What happens if it's 40 degrees outside? How much sport do you want to do?
No.
So if you noticed how, if we're starting off at 0, and as we're going hotter, it starts increasing in photosynthesis up to a certain point, right? And then it gets too hot, and then what happens? It starts decreasing. Okay, so don't forget it. That's the first thing. Second of all, we've looked at light intensity, right? What about how much water? Okay, no matter how much light you put in, if it's not enough water, it's not going to happen. Right, so let's see. What is it? Come on. Okay, increase the carbon dioxide, and of course, the water and the heat. Very temperature and water. Very nice. Okay, and then the last one for this is, what conclusion can you arrive at this whole point? What is the whole point of it? It is, as the light intensity increases, as there's more light intensity, more oxygen is being produced. If more oxygen is being produced, more photosynthesis. Right. Let's have a look. Light intensity decreases, the photosynthesis will decrease. If light intensity increases, photosynthesis will increase. Right. So that is the whole thing. Now, it's almost time to go. Right. And I know I haven't answered any of your questions, and I do feel bad, but help your friends out. If you see them there, hey Looney, they see them on there, help them. I don't know you guys are
Help
clever
your
enough.
friends.
Hey, help your friends and have fun.
Yes.
Hey Looney,
Yes.
all yours, girl.
All right. Thanks.
Pleasure.
Thanks, guys, for tuning in. Remember, if we we didn't get through all your questions, so please send them through the help desk. One of them is learn.mindset.co.za/help desk. Send your questions through there, and you will be answered. But from us,
For a
until
long time.
I was just about to say, until we see you again, like for the longest time,
I'm going to miss you.
we love you. Goodbye, guys.